African Academic Diaspora Virtual Homecoming 2020. The Association of African Universities, AAU, in collaboration with the Government of Ghana and the African Union, is inviting all educational stakeholders to the first virtual AAU Academic Diaspora Homecoming. The overall objective of the homecoming is to strengthen collaboration and partnership among research and education institutions of higher learning in Africa and the African diaspora to advance quality of education and general well-being of all persons of African descent. The main theme for this event is Advancing African and Diasporan Academic Relations. The date is September 28th to 30th, 2020. Time is 15 o'clock GMT. Kindly convert the time to your local time. The venue will be the Virtual Association of African Universities e-Forum, with the registration link on your screen. For more information, contact us now, namo@aau.org or secgen@aau.org. You could call us or WhatsApp us on +1312 539 8473 +233 243 298 464.
Hello everyone. Here we arrived to the last session of our conference, the first International Pharmaceutical and Medical Conference in response to COVID-19 under the theme of Salute Healthcare Heroes. And in this session we have a variety of eminent professors and speakers. We will share their experiences and we will have good news by the end of the session. Please remember to be ready for the group photo by the end of the session and to share with us the recommendations of our conference. And I'd like to introduce our dear professors who will moderate this session, Professor Samah Lutfi and Professor Iman Hassan. Professor Iman Hassan is Vice Dean of Student Affairs in Faculty of Pharmacy, Al-Azhar University. And I'd like to welcome her to introduce our guest moderator, Professor Samah. Please welcome Professor Iman. The platform is yours.
Thank you, Professor Amani Sharif. Welcome in the second session of the final day of Al-Azhar First International Conference in Pharmaceutical and Medical Sciences, a response to COVID-19. This conference is hosted by Faculty of Pharmacy for Girls, Al-Azhar University, under the theme of support for healthcare heroes. I'm honored to introduce our moderator this session, Professor Samah Ali Lutfi. Professor Samah is a virologist and a dedicated scientist. She had a huge, astonishing experience working in several national and international fields. She is the professor of virology and immunology in Cancer Biology Department, National Cancer Institute, Cairo University, and National Technology Research Center, BUE Egypt, Executive Manager for Training Program on Infection Control at NCI Cairo University. Also, she had a great history working in several countries and under several distinguished institutions, including but not limited to WHO and several universities and scientific programs in several countries. Welcome with us, Professor Samah Lutfi.
Thank you very much, Dr. Iman. Thank you very much, Dr. Iman, for your intro. Thank you.
Thank you. Now, we would go for the first speaker. Our first speaker today is Professor Our first speaker today is Professor Ravindra Bhatt. He is a dedicated scientist with a distinction several awards, actually, multiple awards winning work. And he is also a passionate teacher with long experience with many students. He had also been working in several institutions and entities, dedicating much of his time for science. Please, Professor Bhatt, the virtual floor is yours.
Yeah, thank you very much. I will start my presentation. Okay. Am I audible and visible? Is the presentation visible?
Yes, Professor, go ahead, please.
Yeah, thank you very much. I must thank the Al-Azhar University for its wonderful things that they're doing. I'm really impressed, and I want to share some of my thoughts in this regard of COVID. This takes me back to my school days when we saw a story where blind people wanted to know what an elephant is. They wanted to know the elephant, so they requested someone, show me the elephant. They can't see. So the person took them there and they all tried to feel the elephant at the same time. And one said, this is like a wall. Other said, this is like a rope. Other said, this is like a spear. Other said, this is like a tree. They started fighting. Each one was partly right, but all of them were wrong. This is the story of a very big elephant. Now we have an invisible enemy in front of us, something we can't see even with a good microscope. And it is no wonder that we are all blind to the entire part of the enemy. We don't know exactly what it is. We know something, we know it has a spike, it is coming. There is a very specific lock in our system in certain organs like lung, intestines, kidney. There is a ACE2 receptor. This is a beautiful lock, very intricate lock. This enemy is coming with spike protein, is a key ready for that lock. Just imagine it is coming with a key, perfect key for that lock. It opens, it gets in to the receptor, opens the lock and comes. Out comes its RNA, and there is an enzyme to make it stable. It has come ready. Then it has come ready with certain proteins to allow it to enter the lung cells, multiply there and come out. When they come out, they destroy those lung cells. The lung cells die, and these will enter other cells, or they come out and enter the air, and from there they go to other human beings through airborne and through the droplets. So just suffices to say how sophisticated.
Excuse me, Professor Bhatt.
Yes.
Your background vanish your face completely. I think it's better if you disable the virtual background because we cannot see you, only see the bridge.
How do I do that?
From the bottom near to the start video, you just unclick, choose virtual background.
1 minute. How do I unclick that? No, it is not clicked, madam.
Not working.
It is not clicked.
What?
No, it is not clicked.
Hello? Not working?
Yeah, not working. Can I continue?
We cannot see you actually.
I don't know how to come out of that.
Yeah. Okay, can you share your screen?
Yeah,
Okay.
I will share. I don't know what happened. I'll enter again.
Professor Bahat, would you please share your screen?
Something is going wrong. I'm not able to do it. Okay. Can you, something is blocking it. Okay. Can I leave and re-enter? Something is blocking me. I cannot continue. Okay. Can you hear me? Can you see me?
Yes, we can hear you, Professor, but what we see now is the virtual background, or maybe the profile
No,
picture
it's a
you are
virtual
using.
background, but I cannot put the presentation. It's not going.
We will try to share your presentation from our side.
Okay.
Wait for a moment, please.
Yeah.
Can you leave and join again, Prof?
Yeah, I will do that.
Sorry for that. While waiting for Professor Bahat to rejoin us, I'd like to remind you the tracks of our conference. The first day was mostly under the theme of healthcare system. The second day is concerned about the impact of the current pandemic on education and technology. We had several valuable talks in these 2 days. Today, we are talking about the impact of the pandemic on environment and economy and industry. We also are having talks discussing the interaction between environment, industry, and technology with the pandemic, meaning that sometimes we are going to talk about how the pandemic had an impact on the environment and industry. And on the other hand, we would talk about how the pandemic is affected by the environment and industry. And now we are waiting for Professor Bahat to join us. I hope the technical problem will be solved soon. Kindly, please don't forget to fill in the questionnaire. We would appreciate any recommendations, any advices, every suggestion for improvement would be appreciated. Also, any scientific contribution would be is most welcome. Dr. Bahat, please host, could you please make Dr. Bahat a co-host to access the share screen option? Yes, welcome back, Dr. Bahat. Professor Bahat? Professor Bahat, you can now share your screen. You are a co-host now. You may rejoin the video and share your presentation, please. Welcome back.
Okay, can you hear me, madam?
Yes, your voice is very clear. Go ahead,
Very
bro.
clear. Okay.
Go ahead.
I will speak. No video is needed. I will speak. Please remove that. Okay. This is the enemy that we can't see. This has got a beautiful key for an intricate lock. The spike protein, it enters the lung cell, and when it enters the lung cell, it is accepted there, and the RNA comes out, multiplies, comes out, and the multiple copies are made, and the vesicles are born, and they just come out of the lung cell, destroying the lung cell. So when it comes out of the lung cell, two things can happen. It can infect other cells, can come into the air and infect the other human beings. Now, what we did not know is that there are normally a disease has two phases, a phase of viremia or bacteremia and the phase of clinical features and complication. This has three phases. It has got an early infection phase where the patient may be in incubation period or may be completely asymptomatic. And it has got a second phase, which is lung phase or pulmonary phase, and then there is a critical phase. See, what happens is initially there are viruses, but no symptoms. That's when they start spreading the virus. And then there is a phase of viremia and the symptoms come. This prefers the lung cell. So the most important features are in the lung. And then comes the human host response. The host reacts in a certain way that sometimes nothing much happens. Sometimes there is a very severe response. And the severe response is not only in the form of stimulation of the T cells and B cells, it also stimulates the situation of hypercoagulation and stimulation of the neutrophils and stimulation of the macrophages. That means the immune system is responding more than what is required, and instead of killing the virus, it's now damaging the human tissues. This is something unique. This is called cytokine storm. This happens not only in COVID, it happens in any antigen antibody conditions. It happens in SLE, we call it flare. It happens in rheumatoid arthritis, we call it flare. But what happens here is that there is difficulty in breathing. The person becomes breathless and the person has low oxygen. And it is a low oxygen that can kill the patient. So initially, we did not know the reason for low oxygen. We thought it is due to either infection from the virus or infection from the bacteria. But now later we understood there was a situation where a frustrated, this I got from New York from one of my students. The frustrated pulmonologist met a frustrated nephrologist. Pulmonologist said, my patients have hypoxia. I give oxygen, I ventilate, they die. The nephrologist said, I have people requiring dialysis, but I can't dialyze because their vessels are blocked. Both agreed that it is not oxygen is not going, not because of pneumonia, but because of the blockade of the vessels, thrombosis. So it took us 3 months and thousands of deaths to realize we are on the wrong track. And it was required not only to give oxygen, not only to give antibiotic, but also to give antithrombotic drugs, anticoagulants, and ventilation to be reduced. So hypoxia had to be, we had to find out why there is hypoxia and address to the problem. And what happens in the lung vessels also happens in other vessels, heart vessels, and also in other vessels. So much so, in the lung it is hypoxia, in the heart it can produce heart attack, arrhythmias, myocarditis. So what we thought is a disease of the lung only, no, it is not a disease of the lung. We were wrong all the time, like the blind man. It also involves other structures. It also involves others because it involves vessels. If the lung cell or the ACE inhibitor on the lung cell is one thing, such receptors there in intestine, kidney, heart. Also, it involves the endothelium of the vessels producing thrombosis and blocking all the vessels. So you have a situation where multiple organs are involved, and therefore it is a systemic disease. So you have a situation where there is cytokine storm damaging multiple vessels, severe disease. We were desperate in the first 3 months, putting them on ventilator, and we were losing. 80% of the people who were beyond the age of 70 never came out of the ventilator. This was bad. There were thousands of deaths in United States, Italy, and Brazil, and other places. So what really happened, there were a lot of players in the game, like interferon, tumor necrosis factor alpha, interleukins 1, interleukin 6, interleukin 18, but it was not only the interleukins, it was the entire process of inflammation. Then we started experimenting on the drugs. What drug would be best? Initially, we realized that hydroxychloroquine and the chloroquine block the entry of the virus, RNA destabilized by remdesivir or lopinavir. Then we realized that hyperimmune response is the killer in the second stage, and that's when they die even despite the ventilator. And we realized that the blocking this interleukin and other things will help. So tocilizumab came into picture with the interleukin 6 blocker. We were blocking all the things. I shown a picture of a tree here. There is a tree. The tree has stem, the tree has leaves, it has branches. We were going branch by branch. We were giving interleukin 6 and tocilizumab. We were giving different things for different situations. But what we didn't realize is, if their leaves are toxic, why don't cut the tree? And that we realized only after the recovery trial in end May, early June. The report started coming, and then we realized that dexamethasone is a wonder drug for this, because it cuts the stem itself of the toxic tree. It cuts the stem, and therefore it is the best thing to do. That is the give dexamethasone. Going a step further, when the death started coming down at the late stage, early stage, you give budesonide inhalation, much better results. Fewer people will go for this late stage, much better. So we realized from the studies looking back in Italy, out of 65,000 people who died, only 20 patients were rheumatology patients, meaning they were either on hydroxychloroquine or on steroids, and their course was much better. And therefore, with a gamut of drugs, whichever is available now, we are able to choose. Now, I want to show a very unusual presentation. This is what happened in March. This I'm taking a random patient. Patient was admitted on 2nd March, and the X-ray was normal on 5th March, and he was tested for COVID positive on 6th March, and given Caletra, lopinavir, ground glass opacities came, and Caletra was didn't seem to work. They did CRP and procalcitonin. It was increasing, and they gave antibiotics, ventilated him, and then he improved. Now the question is, he went into shock, and then he improved. The question is, if this is in March, you don't see some drugs here. You don't see the presence of dexamethasone. You don't see the presence of heparin. You don't see the presence of aspirin, because we did not know. So later, if this patient had come now, what would have been the drugs? We would have definitely given him antiviral treatment in the beginning. then anti-inflammatory treatment, we would have started on methylprednisolone or dexamethasone or hydrocortisone, depending on the physician's choice. We would have given him enoxaparin, and we would have reduced the chance, we would have reduced the requirement for ventilation. So I recommend, and what I believe, we should learn from our experience, the past. We should document our each patient experience, something like this with the date and the drug. And when we look back, we know where we went wrong. All of us are in the learning phase. No one is perfect. You saw my presentation. We are all in the learning phase. However senior, we learn. And this disease is a great teacher. It makes us respond to emergencies. So this is how I learn. This is how I recommend anyone to have a chart. This is a basic chart, simple chart. Put the date, put the drug, put the response. And now looking back, great heroes are missing. No dexamethasone, no heparin, no presence of major drugs which are used now. This is why mortality was more. And Dr. Islam before me said mortality is coming down. Yes, because we are knowing the drug, we are knowing the disease better. So we use antivirals, inhale budesonide, hyperimmune response is corrected. So we don't allow the disease to go further. And we have a vaccine. Do we have a vaccine? Well, it's a very difficult question to answer. The very easy concept, a vaccine presenting a part of the virus dead or alive, antibodies are produced. We hope that antibody works against the spike or the RNA, and the T and B memory cells remember the enemy. There are many players in the game. There are many companies in the race, but the question is, will we ever have a virus? Because this enemy is intelligent. An average virus, RNA virus, it is double the length of an average RNA virus. And because it is double the length, it has more genes. Because it has more genes, it has more mutations. Because it has more mutations, it can change very fast into different structure. So which one we are targeting with the vaccine? If it's RNA, that will change. If it is the genes, that will change. So getting a success out of the RNA is not a certain dream. It's a dream, but we don't know whether it will become reality. So we also use repurposed drugs. We are using ivermectin, doxycycline. We are using vitamin C, vitamin D, and we are now happy about inhalation steroid, dexamethasone against inflammation. We also find children do not get much severe disease as much as in elderly or adults because probably they have thymus, and sometimes the fever can last longer. So what do we learn from this? What in this context, what do we learn? We learn that the old things, if we are alive today, this is because our ancestors withstood the rampage of pandemics like plague, the pandemic like influenza, all the pandemics they withstood. That's why we are there. There was no drug at the time. There was nothing at the time. They lived because they kept themselves clean and avoided the patients, which means using the mask, washing the hands, using the tissue, social distancing. These are still the main things. Now, what about the knowledge in the field? We are very confident in the beginning, high confident. We said we know everything, and then we realized we know nothing. People were dying. Then we started trying to make sense. Today we are making sense. But what we have become more humble. We now know it's very complicated, and the knowledge in the field is increasing, but we are becoming humble. We are still learning. I end my presentation thanking the audience, thanking the organizers, and saying, putting this slide once again, let us learn that we don't know, and let us keep our eyes and ears open. Let us learn from each other. Let us learn from our patients. I think a time will come soon when we understand the disease, not by things that we don't have. We don't have a vaccine. We don't worry about it. We practice what we have. We understand the disease. We use what we have. I'm sure we will do much better. And with this, I thank the organizers and thank you very much, everyone. Thank the audience. I'm sorry there was a technical hitch, and I end my presentation with this. Thank you very much. If there is any question, I'll be glad to answer. Anything, any questions are there, madam? Sama.
Thank you, Prof. Bahat. If anyone would like to ask Prof. Bahat, you can use a chat box, and please, Prof, keep being with us till the end of the session.
Sure.
Okay. And
sure, I will.
yeah, okay. And now, Prof. Samah, kindly introduce our next eminent speaker.
So I'll stop sharing the screen. Thank you very much. I'll remain connected.
Thank you.
Thank you.
Dr. Samah,
Prof. Samah, can you unmute your mic? And Professor Dr. Tariq Al-Arabi is already here, so I would like if you can introduce him, please.
The voice clear for you?
Yes,
Can you hear me? I would like to introduce Professor Dr. Tariq Al-Arabi. He is manager of UNGB, Medical and Electronic Waste Management.
The voice is not clear, by the way, Dr. Samah. Okay, I think we can solve it while Prof. Tariq give his speech. And if you allow me, let me introduce Professor Dr. Tariq Al-Arabi from the Environmental Agency. Sorry, Anas, can you add the slide again?
Professor Tariq Al-Arabi is the manager of GEF.
Maybe my team get tired, I don't know. But anyway, we have Dr. Tariq Al-Arabi right now with the presentation to reflect and give us feedback how the Environment Authority and agency, Environmental Affairs Agency, have impact on our health. And let's introduce the platform is yours, Dr. Tariq Al-Arabi, to introduce your talk.
Thank you.
And I'm sorry if the talk is written in Arabic.
No, it's in English. Just one slide or something like that. Can I share my screen again? Okay.
Yes, please, Professor.
Hi, good afternoon for everyone. My name is Tariq Al-Arabi, not Al-Arabi, but it's the same, don't worry. I will represent the effort done by the Egyptian Environment.
I'm sorry for that, Prof. I'm sorry for that.
No problem, no problem. I will represent the efforts done by the Ministry of Environment and Egyptian Environmental Affairs Agency in the field of healthcare waste management, and especially during the COVID-19 pandemic. First of all, we are, as you know, how can I? First of all, you know that the Ministry of Environment dealing with the waste. So our concern is the effect of COVID-19 pandemic on the medical waste, and of course the way of treating this waste and how it will affect the environment. First of all, we start with a baseline assessment of the healthcare facilities that we have in Egypt. This was done before the pandemic, of course, and one of our concern or our findings is that we find that all healthcare facilities in Egypt are just segregating the healthcare waste into just three streams. One of them is the general waste, and the other is the infectious waste, which they put in the red bags, and the sharps, of course, that they put it in the safe box. But they ignore the chemical wastes and the drugs and cytotoxic drugs and also anatomical waste and any other waste that contain PVC plastic. PVC plastic is the polyvinyl chloride plastic. And we find that there is improper segregation of this waste, that we find that approximately 12 from 13% to 40% of general waste bags, we find it that it's containing hazardous waste, and which is very dangerous because the persons dealing with the regular waste is not trained to deal with the hazardous waste. And also we find that approximately more than 50% of the infectious waste bags having regular waste. And we find that the percentage of the medical waste is very higher than the international norms. So we find that from 51% to 71% of the waste coming out from the hospitals are in healthcare waste. And the norms, the international norms, is around from 10 to 25%. This is a picture showing what we found, that this is a red bag that should have the medical waste. We find pieces of wrapping of chocolates, and we find, for example, sharps in the red bags, not in the safe boxes. So we refer this observation to the lack of information and training of the healthcare staff in the healthcare facilities, and also that they are classifying non-infectious waste as infectious waste. For example, the bottle of saline solution, for example, they treat it as an infectious waste, although it is not infected by any diseases, any viruses or bacteria, because it's not touching any of the fluids coming out from the ill peoples. Also, this high percentage of hazardous waste that we find it, this reflects on the raising the cost of waste treatment, and of course the substandard treatment of the medical waste itself. So we and by the end of our assessment, we use a checklist which is done by WHO and the Global Environment Facility. So we find that this model healthcare facilities that we assess getting a rate from 44% to 76%. So, and also this is what we mentioned before is inside the hospitals itself. And on the other hand, the way of treating the medical waste, we find that most of the incinerators used is substandards. Also, they are consisting of two combustion chambers, but there is no air pollution control units. So this make us to believe, based on, of course, on the emission factors calculated from this incinerators, that we have an air pollution from 3,000 to 40,000 times the permissible limits stated in the international and national laws. This limit is for the POPs, which is the persistent organic pollutants, and this is the main concern of the Ministry of Environment to reduce this POPs emission. And also we find that in some cases, the available treatment facilities are not sufficient to treat all the hazardous waste coming out from the hospitals. So we make an intervention plan. Sorry, this is the only one in Arabic, but it has a translation in the next slide. So we start to make plans and guidelines and policies for the healthcare waste facilities, and also to propose reformation of the employment structures that they have. So one of our interventions that we said that we must do it is, of course, the training, and to have responsibility to dedicate waste management officers in all of the healthcare facilities, and to accredit this waste management officers through exams after they took this training materials that we already have it from the WHO and from the Healthcare Without Harm. The training also will be used, of course, it will be reflected on the good separation or segregation of the waste, which will be reflect, of course, on the amount of waste that will be generated. And of course, we can implement the fair price for treating this waste, because the price that we have it now in Egypt is very low, and because of this we have a substandard treatment of the waste. And also if we implement the fair pricing, this will encourage the private sector to enter to this field and to invest in it. Also, a good separation is very crucial in the way of treating this waste. For example, if we are now promoting for shredding and autoclaving. So chemicals and drugs and cytotoxic drugs, we cannot treat it using autoclaving, of course. So we must separate these two streams from the beginning. And after treating with autoclaving, we should have it, we should from beginning, we have the consumables, the medical consumables free from PVC, to be able to use the output of the autoclaves as an RDF for the cement factories. Also, we propose to have a reforming of the decrees that they have for the financial stability of the system itself, to be able to sustain the training and the supplies used for the medical waste segregation and treatment. This is the English version, a summary of what I already discussed in the last slide. And also one of our proposals for encouraging or for making a good system is to encourage the private sector, like I said, to enter to the market. And also we offer them funding for the Ministry of Health, to have this fund to replace the autoclaves to replace the incinerators with a new autoclaves. This is what the guidelines and the policies and the plans that we already issued during the last period. And this guidelines help them a lot during the COVID-19 pandemic, to show them how to transfer the medical waste and how to temporarily store them. And we already construct a website for the healthcare facilities to register their amount of waste that is generated daily, and the timing of transferring this waste to the treatment facility. And also the treatment facilities, they have an access to this website to show us the quantities that they already treat and what they did with the output of the treatment options. And also we put in our guidelines the different waste streams that we will deal with them. We separated into 6 categories to be able to treat them using the facilities that we have it now in Egypt, the substandard incineration, like I said. And for example, these substandard incinerators, we cannot burn any PVC on it. And also for chemical and pharmaceutical drugs, we must not use the autoclaves for treating it. So we can use, for example, solidification for cytotoxic drugs and burying it in hazardous waste dumping sites. Thanks a lot for the invitation, and I hope that I cover some of the activities done by the Ministry of Environment during this period. Thank you. I'm ready for any question if you have.
Thank you very much, Professor Tariq Al-Arabi, manager of the Waste Management Project. Thank you for the much needed lecture. Actually, when there's a pandemic running, your speech is much needed. One question for you, please, if you please answer. Are these treatments mean recycling?
No, there is no recycling for medical waste. Treatment, we have 2 ways of treatment: incineration or autoclaving. For incineration, the output of the incinerator is just a little bit of ashes, which should be dumped in hazardous waste dumping site. For the autoclave output, if it is separated in a good manner in the beginning, so which I mean, if it's not including any chemicals or cytotoxic drugs, the output of the autoclave can be treated as a normal solid waste or municipal solid waste, and we can use it as RDF and use it as a fuel for cement factories. But recycling is prohibited here in Egypt for medical waste, because we are afraid that there will be a reuse for this medical waste. So we shred it and destroy it completely.
Another question, please. What about going to cement factory?
What about going to cement factory?
Cement factory, yes.
The output of the autoclaves, it has a high calorific values, because we know the medical supplies is rich of plastic and other items with high calorific value, so they can use it as alternative fuel in cement factories. But this on a condition that it is well segregated before it enters the autoclave.
Thank you, Dr. Tariq Al-Arabi, for the valuable speech. Now, our next speaker is an infamous professor, Professor Ashraf Abbadi, the head of Pharmaceutical Chemistry Department, Faculty of Pharmacy, German University in Cairo. Also, he was the former dean of the same faculty. He had great experience working in several countries: Egypt, USA, Saudi Arabia, and Germany. This way, he had an interesting scientific record. Basically, about medicinal chemistry, drug discovery, and computer-aided drug design. This last one is much needed these days, and it's actually one of my favorites. So with no more hesitation, Professor Ashraf Abbadi, the floor is yours.
Thank you very much, Professor Iman, for the kind introduction. Also, I would like to thank Professor Amani Sharif for inviting me to this important conference. Today, I will start to share the screen.
Yes, please. Go ahead, please.
So today, my talk is about drug repurposing in the era of COVID-19. The outline of the talk, it comprises definitions and backgrounds about COVID-19, advantages and strategies of drug repurposing, and some examples of repurposed drugs. So what is meant by drug repurpose? It is mean profiling of an approved drug for the treatment of a different conditions or disease than that for which it was originally developed. So this is what is meant by repurpose and sometimes named drug reposition. As you know, by March 2020, the WHO declared coronavirus or COVID-19 as pandemic. So what is meant by pandemic? It is a disease affecting a large proportion of population occurring over a wide geographic area, such as a section of a nation or the entire nation or a continent or in the whole world, as in the case of coronavirus. Actually, the word pandemic comes from the Greek word pan, which means all, and the word demos, which means people. It means collectively pertaining to all. So this is what is meant by a pandemic disease. To understand the importance or how this COVID-19 affected our lives, we should talk about 2 points: its infectivity or contagiousness and its fatality. So if you look to this red square, you can see here that the average number of people infected by each sick person is around 3. So actually here, each infected person with COVID-19 can infect up to 5 persons, and the average is 3 persons. However, its fatality rate is not that high. Its fatality rate is around 4%. So this square represents the infectivity and the fatality of COVID-19. As per fatality, for example, it is much less than Middle East respiratory syndrome and much less than SARS, which are also coronaviruses. They are caused by coronaviruses. So now we'd like to talk about the principle of drug repurpose and why did we refer to this one in case of COVID-19. When COVID-19 occurs, actually there is no approved treatment for COVID-19 infection. There is no treatment for this infection. As an emergency, we don't have the time to develop a new drug from the scratch. Actually, you have to look only for the tools you already had. So the magic solution in this case is drug repurpose. Since we are repurposing known drugs for new indications, so at least at the beginning, those repurposed drugs should be used mainly in hospitals and under close medical supervision. Number 2, there is a difference, not to mix up, there is a difference between what is known as drug repurpose and off-label usage. The main differences between the two is that the drug repurpose is more linked to clinical trials and authorization by the respective health authority. So this is the main difference between repurpose and off-label usage. What are the advantages of drug repurpose? First, you will decrease the time, cost, and uncertainty about the development of this drug. Number two, you are going to reduce the number of required clinical trials. So actually, if you would like to make a clinical trial by this repurposed drug, you can start from phase 2 or phase 3 clinical trial. You don't need to perform phase 1, or of course, you don't need to perform preclinical development. So by this, you are reducing the time and the cost of the medicine to reach the market for the new indication. Number three, existing pharmaceutical supply chains could facilitate formulation and distribution of this repurposed drug. Also, you should have a clue, just a clue about the possibility of combining this drug with other drugs in order to potentiate or synergize treatment. So, what are the intervention points in case of COVID-19 infections? Actually, in case of COVID-19 infections, you should think of 5 words. Number 1, the virus. Number 2, the immune system. Number 3, the CNS, and I mean by this, the fever developed due to the viral or secondary bacterial infection. Number 4, the effects of the virus on the cardiovascular system, particularly blood clotting. Number 5, the respiratory system, and I meant by this what is known as respiratory depression or ARDS, acute respiratory distress syndrome. And by the way, the effects of the virus on the cardiovascular system or on the respiratory system are the main causes for death. So again, we need 5 words whenever we are thinking about COVID-19, and we are going to repurpose drugs and intervene in these 5 points. Now we'll talk about repurposed classes. Of course, you should think of antiviral drugs. For example, you can think of using those drugs which are active against viruses from the same family in search for what is known as cross-activity. So if we know that MERS and SARS are from the same coronavirus family, so why not to use drugs which are active against MERS and SARS, or those already active against seasonal influenza for the treatment of coronavirus. So this is antiviral drugs that are going to be repurposed. So this is number 1. Number 2, you should think of immunomodulatory agents. And actually, you should think of these immunomodulatory agents in 2 phases. The first one at the beginning of the infection or before the infection. In this case, you are in need of repurposed drugs which will strengthen the immune system. But whenever there is a strong reaction between the virus or the accompanied bacteria and the immune system, in this case, you will need of repurposed drugs to suppress the immune system. So actually, for example, at the beginning of the infection, in order to prevent or minimize the symptoms at the beginning of the infection, you can look for immune system boosters. For example, these boosters, for example, like zinc, vitamin C, and so on. But in case of this severe reaction between the immune system and the virus, you need to suppress this reaction. And in this case, you can look, for example, for dexamethasone, cortisone, or interleukin 6 neutralizing agent, for example, tocilizumab. So here you have like two ways of thinking. Also, you should think of drugs which prevent blood clotting, because we said that its effect on the cardiovascular system and the blood clotting will lead to organ failures and may lead to death of the patient. So among the drugs which were repurposed for prevention of blood clotting and used in COVID-19 infections, we have aspirin, rivaroxaban, and heparin. Also, you should think of preventing respiratory depression by blocking the cytokine storm. And this cytokine storm again results from the reaction between the virus or secondary bacterial infection and the immune system. And this will lead to huge release of this cytokine storm. And this can be suppressed by dexamethasone, which is a cortisone derivative, or tocilizumab, which is a monoclonal antibody. And in final, you can think, of course, of using antibiotics, for example, paracetamol in case of fever. So what are the strategies of repurposing? How we think of repurposing a drug? We have four strategies. First one, previous experiences and literature search. So this is number one. Number two, computer-aided drug design. Number three, block of disease pathogenesis and manifestations. For example, the effects on the cardiovascular system and the effects on respiratory system. Again, you are in need of clinical trials, even if it's small in size. So I'm going to show you now applications to these strategies in each case of repurposing. First strategy, search in literature for drugs which are active against viruses from the same family or close family, looking for what is known as cross-activity. For example, favipiravir, it is the Japanese drug. Actually, this drug was mainly developed against seasonal influenza. It is polymerase inhibitor of seasonal influenza. But since influenza is RNA virus and its symptoms is similar to that of COVID-19, they thought of using favipiravir for treatment of COVID-19 infections. That's how it was repurposed, and now it is approved for treatment of COVID-19. Number two, remdesivir. Again, remdesivir is polymerase inhibitor. Mainly, it has been tested against the viruses causing SARS and MERS, which are from the same coronavirus family. So why not to repurpose it for the treatment of COVID-19? Now it is officially approved by the FDA for treatment of COVID-19 infections. Even the antiparasitic ivermectin. This ivermectin actually is known from before that it is active against the integrase enzyme of HIV. So HIV, it's RNA virus. So why not to use this ivermectin in case of SARS-CoV-2? So this is how it comes to be a drug in the protocols used for the treatment of COVID-19. Even if we look for hydroxychloroquine, hydroxychloroquine is mainly anti-malarial drug, known to block the viral infection by increasing the endosomal pH, and this will prevent the virus cell fusion. And it was found to be active in case of SARS and MERS infections. In addition, in February 2020, it was found to be of in vitro activity against SARS-CoV-2. And in March 2020, there was a small-sized clinical trial that showed that for those patients with positive PCR, here you can see if they got a placebo, there is no suppression in the number or in the PCR titer of this COVID-19 infection. In case of hydroxychloroquine, this is the blue line, there is a reduction by 60% in the titer of this virus as determined by PCR. And if you combine this hydroxychloroquine and azithromycin together, this will lead to full recovery, although the number of patients was just 6. But this will give a clue that this combination of hydroxychloroquine and azithromycin is a useful combination. Here you can see some of the known drugs which are known to stimulate the immune system. So actually, in order to prevent or to minimize the symptoms at the beginning, you can take such vitamins and minerals, for example, vitamin C, vitamin A, B, E, D, and minerals like zinc, iron, and selenium. Also among those drugs developed by repurposing, tocilizumab, Actemra. Actemra is a monoclonal antibody. It is known to block interleukin-6 receptors and mainly indicated for treatment of arthritis, only arthritis. But now, because we know that it blocks interleukin-6, which is a part of the cytokines storm caused by COVID-19, now it is repurposed for the treatment of SARS-CoV-2 in order to treat respiratory depression and acute respiratory distress syndrome. And that's why this Actemra is now among the very effective drugs for the treatment of respiratory depression in case of COVID-19 patients. The same for dexamethasone. Dexamethasone is cortisone derivative. Again, it blocks cytokine storm, and by this, it is useful to reverse respiratory depression and ARDS. Even azithromycin, which we know it as macrolide antibiotic. Actually, here it is not just used as a macrolide antibiotic. It is a macrolide antibiotic and anti-inflammatory agent which reduces the cytokine storm. So actually here it is of dual effect cytokine storm suppressor in addition to being preventing secondary bacterial infection. And here you can see also which drugs were repurposed in order to prevent the clotting and organ failure in case of COVID-19 infections. The most commonly repurposed drugs in this case are aspirin, rivaroxaban, and heparin, and all are like blood thinning agent in order to minimize clotting and organ failures. Another strategy is computer-aided drug design. So after the discovery of the 3D structure of some of the proteins of SARS-CoV-2 and their position in the protein data bank, some researchers started to dock those drugs approved by the FDA into those protein structures and try to look for those drugs that might be inhibitors to those proteins, particularly protease and polymerase. For example, among the drugs which are now in phase 2 clinical trial is the ARB losartan. It is an antihypertensive, but now it is in phase 2 clinical trial for the treatment of SARS-CoV-2 infection because it is a protease inhibitor of coronavirus. The same for chloroquine. It was found to be a protease inhibitor of SARS-CoV-2. Even the anti-hepatitis C virus, sofosbuvir, was found to be active against the polymerase of SARS-CoV-2.
Excuse me,
So
Professor.
now we have seen the different
Professor Abedi,
strategies
excuse
by
me.
which we can think of
Your
repair
time
booster
about to be
drugs.
finished.
We use the literature, we use the clinical trials, we use computer-aided drug design, and all of such things are even combined in order to think how to think of repurposing a drug.
Professor.
By this, I come to the end of this talk. Thank you very much for your time and attention.
Thank you very much, Professor Abedi, for your interesting and informative presentation. And now, can you hear me, Professor?
Yes, I can hear you. Yes, please.
Okay, thank you very much, because I just adjusted my voice. Dear Professor, we have here some questions for you, please. If you don't, I'm sure you are pleased to answer these questions. Here we have a question. Did you try artemisinin derivatives?
Yes.
Derivatives instead of hydroxychloroquine? I think it will be good antiviral agent by drug repurposing process. It is anti-malarial agent also. Also, what about increasing the alkalinity of the body to increase immunity? Thank you. Could you please answer?
So I can answer this very good question. Actually, for artemisinin, it has to be tried. It has to be, actually, if you look to hydroxychloroquine, it increases endosomal pH. This is number one. In addition, it was found to be of activity, of in vitro activity against the virus. So if this is proven for artemisinin, in this case, it can be used. But just for being two antimalarials, I think this is not the main reason to repurpose a drug. So actually, in case of artemisinin, you have to prove first that it is active against a particular protein of this SARS-CoV-2, or it inhibits the replication of this virus in vitro, after which it can be developed, of course.
Thank you. There is another question. Do you think immunosuppressors like this European could help last stage COVID-19 patients?
Sorry, I didn't hear this.
Do you think immunosuppressor like this European could help last stage COVID-19 patients?
I don't know the name of the drug, but let us take it like this. So actually, if you have a drug which is suppressing the cytokines storm, actually cytokines storm is contributing to the blood clotting and contributing to the respiratory failure. So if you have this new drug, which I don't exactly its name, yes, it can be used, of course, if it blocks cytokines storm.
There is another question from Dr. Femi, but it's recommendations. There is need to, 1, develop standard protocols to handling future pandemics, 2, encourage, facilitate institutional-based researches on infectious diseases through adequate funding and industry, academic partnerships, 3, explore what nature has blessed Africa with in terms of the phytopharmaceuticals synergistic. This just recommendations. There is another question. Did you try artemisinin? I think you replied on this. Wonderful presentation. Thank you very much, dear Professor, for your nice and informative presentation. And let us go to the next speaker, please, the fourth speaker.
Okay. Thank you very much.
Thank you very much, dear Professor. Now we will move to the next speaker, Dr. Hala Adli Hussein. She is Secretary General of Federation of Arab Women Leaders, Chairman for Biomisr for Pharmaceutical Industries Company. Dr. Hala Secretary General, Federation of Arab Women Leaders affiliated with the Joint Arab Labor System and the League of Arab States, Chairman for Biochemistry for Pharmaceutical Industries Company, Chairman for Mika Pharma Company, former Chairman and CEO of AG Vaxera, former General Manager of Amoun Pharmaceutical Company, former General Manager of the Development Department of the Farco Pharmaceutical Group, lecturer in the Faculty of Pharmacy Al Mansoura University, member of the National Council for Women from 2000 to 2011, MBA in Business Administration, rapporteur of the Women's Unit Environment Affairs System Delta East till 2011, member of the Women Faith Committee in COPPEM Committee Euro-Mediterranean Partnership of Local and Regional Authorities, member of the National Council of Women from 2001 to 2011. We have several achievements. Dr. Hala will give us a presentation today about COVID-19 worldwide. We will have global idea about COVID-19 situation worldwide. The floor with you, dear Dr. Hala. Sure, we are interesting to hear your presentation. Welcome with you.
Welcome and thank you. First of all, I would like to thank Al-Azhar University for this grateful conference, and it's honorable to be with you, to be with this marvelous talks from several countries. Thanks for Dr. Amani for this invitation, and I'm going to share with you my presentation. I think all the previous lecturer or the previous speakers speak a lot of my presentation, but now I'm going to go through it faster because we are the last speaker. So I'm sorry for your efforts, and thank you again. First of all, I'm not going to repeat all my colleagues have done, that the virus is ineffective agent, and it's inactive until it's going on the cells, and it's not secrete the toxins like bacteria and coronavirus family. They are group related to RNA viruses that cause disease to them in mammals and birds. In human and bird, they cause respiratory tract infection that can range from mild to lethal. But in cows and pigs, they cause diarrhea and hepatitis. And the coronavirus, the history of the coronavirus was discovered in 1930s in a chicken, infectious bronchitis virus, and in 1940s, there were two more viruses were isolated, mouse hepatitis virus and transmissible gastroenteritis virus. The human coronavirus was discovered in 1960s, and there is a very large group of coronavirus, but as WHO, they call there are as yet no vaccine or antiviral drug to prevent or to treat human coronavirus infection. So I'm going to take you in a travel through some countries or some experiments in some countries. In Cuba, they sent to us the Cuban medical doctors. When I asked them, they went to Italy to share in the treatment. This was part of one of the email of the authority. They said that the Cuban medical doctors are until now supporting not just Italy, but 14 other countries as well. They use interferon alpha 2B, which is an immunostimulator successfully applied in China, COVID to patients in Wuhan. This is just one possible treatment used in the world until now. I think it was a successful experiment there in Italy. In China, they use these drugs. When we asked them what the protocol, in mild cases, they use the chloroquine with azithromycin. And in severe cases, they use this traditional medicine. And I sent it to Faculty of Pharmacy of Helwan to analyze this and to know what the herbs they used. I think my colleague, the speaker from India, speak about these herbs. I think there is more than 1 or 3 herbs that he discussed. All these herbs, I think he discusses in India. I think they use the same herbal drugs. One of country used the bee venom. Oh, sorry, there is something wrong. Do you hear me?
Yes, doctor, your sound is clear. Go ahead, please.
Oh, but something is going wrong with the slides.
هي قالت حاجه في الاول مش عايزه تشير البرزنتيشن عشان مش عارفه الوقت.
Me? No, I didn't tell that.
So we can share your
Okay.
Yes, welcome. Go ahead, please, Dr. Hal.
Okay. This is traditional medicine from China. And the bee venom. The bee venom, we use the bee venom in Vaxera. I was the chairman and CEO of Egypt Blood Vaxera. And they use this venom in autoimmune disease. And they made survey was done between February and March on 5,000 local beekeepers, 723 of them from Wuhan.
Dr. Hala, please share your screen. Please share your presentation, please. We couldn't see.
You couldn't see it? Okay.
Yeah, it's not displayed. Please share your screen.
Okay.
Your voice is clear, but the presentation is not displayed.
Okay, I'm going to release. Okay. Sorry. I think it was something is good. Okay, I'm going to really share it again. Is it okay now?
No, it's not displayed yet.
I share it.
Dr. Iman, could we help from our side?
I share it several times. What's wrong? There is something wrong, not from my side, but I should. Did you see it now?
No, it's not clear. It's not displayed.
What side? I'll stop share and re-enter again.
Yes, please.
Okay. Does it now already?
No, it's not displayed.
Something is going wrong, but not from my side, sorry. Okay, I can go from without the screen share.
Or you can, please go ahead.
Okay. The bee venom was a very successful experiment in Wuhan, and it was applied also in SARS and MERS. They discovered that they enhance the differentiation of human regulatory T cells, which play an important role in control SARS-before. So, as Dr. Ashraf said, they repurpose some of the drugs, like hydroxychloroquine, favipiravir, remdesivir. Also, we use here in Egypt, in some cases, sofosbuvir and daclatasvir for 14 days with pharco experiments. And dexamethasone also was very successful, especially in pregnant women. The doctors advise the pregnant woman to take from 4 to 6 dexamethasone before delivering babies to enhance and make sure that the lungs doing well. So the treatment here divided in the purpose or the basis of treatment of coronavirus or any virus depends on 3 things. First of all, is immunoregulator drugs, immunosuppressive. On surveys with doctors with kidney transplantation, they tell us that no one of their patients was infected with COVID-19 as they take tacrolimus as immunosuppressive. The BCG vaccines. There is one paper said that BCG vaccination policies make a 10 times difference in COVID-19 mortality. New study was held in New Delhi. But there is some precautions when we give the vaccines, the BCG vaccines, as we have to make the tuberculin test before giving the vaccine, because it's very dangerous. So we have to give awareness to all the people for this. The convalescence plasma therapy was approved by FDA, by WHO and CDC in Ebola. So it gives us a very marvelous results. We cannot approve, but it's already used in treatment of Ebola before. So the developing of vaccine. Any vaccine developing take from 18 to 24 months. So all the social media takes that there is a vaccine finished. No, not yet. From 18 to 24 months, this is a clinical trial. Some of the companies make clinical trial parallel when they assure that the results from the second phase is okay. They started the phase 2 in parallel with phase 1 and phase 3 in parallel with phase 2, but they have to complete all the phases. So there is not yet vaccine already. There are some drawbacks from vaccines like cardiovascular vaccines, rashes, and very various skin rashes. One of the trial, well, not trial, there is an instrument. This instrument is, I would like to share with you the photos, but I can give it to Dr. Amani later. The instrument was registered by our military. It's patent for them. It's discovered from 2012. And it was honorable that I one of the team that evaluate this instrument, and one of the clinical, one of the committees that tried this instrument in Vaxera on more than one devices. It's tested for the viruses, virus C, virus B, AIDS, malaria. And I think they can separate the corona and make this an instrument for just test the viruses, even if on the wall and the instrument, and it is tested in all Vaxera instruments when I was in Vaxera at the time. And there is a thesis held on this instrument. I think it will be marvelous, and it will be a very good discovery. And it is patent to military, Egyptian military, and you can see it on Google. Just write CFAST instrument, or in Arabic جهاز الكشف عن فيروس C, أو ال C fast in Arabic or in English. You can show the publications, you can show the thesis that more than 4 theses on this instrument. I think it will be very good. I'm not going to talk more because my colleagues talk enough about the disease, about the plasma, about the PCG, about the clinical trial which is going on. And thanks again, Dr. Amani, for this invitation. And I let the floor or any question if you want.
A lot of thanks, dear Dr. Hala, for your nice speech, and hopefully we can see your presentation later. I wish if there is any question to Dr. Hala before we move to our next speaker. Is there any question for Dr. Hala? Here, is there any studies done on medicinal herbs in Egypt? Which one of the antiviral gave more successful results than chloroquine? Could you please answer this?
I am sorry, there is no regulation for herbal registration in Egypt unless we register it not in MOH or on the EDA now. No guidelines for still approved for herbal registration as a medicine now in Egypt.
Yes, thank you very much. There is another question, but I think this is chloroquine have antiviral effect or only reduces cytokines storm?
No, it's immunoregulator.
Yes.
Just immunoregulator.
Okay, thank you very much, Dr. Hala, again for your nice talk. We can move now to our next speaker because the time is so tight with you, Dr. Iman. Can we present the next speaker, please?
Thank you, Professor Hala Adli. Thank you for the valuable presentation, and we are sorry for the delay. Now I'd like to present our next speaker, Professor Dr. Gamal Al-Afandi, Associate Professor of Environmental Sciences, Tuskegee University, USA, ex-Director of International Research and Development Project, and the same university, Professor of Climate and Environment, Department of Astronomy, and Faculty of Pharmacy, Faculty of Science, Al-Azhar University. Professor Gamal is interested basically in the relationship between environment or the interplay between environment and human health. And he has great scientific records in this field. So let's start Professor Gamal Al-Afandi's talk for now. Professor Gamal?
Bismillah ar-Rahman ar-Rahim. I'd like to take this opportunity to thank you for your invite and the excellent organized conference. In fact, I am honored to be invited as a keynote speaker in the first international online conference, Pharmaceutical and Medical Sciences, a response to COVID-19 in support healthcare heroes during September 1st to 3, 2020, which is organized by Faculty of Pharmacy, Girls, Al-Azhar University, Egypt, in collaboration with the Regional Office of Association of African Universities and the Egypt Knowledge Bank. My extended gratitudes and sincere thanks to the following professors: the conference chairperson, Professor Dr. Amani Sharif, Dean, Faculty of Pharmacy, Girls, Al-Azhar University, conference chairpersons, both Professor Dr. Sahar Ramzi Radwan, Vice Dean for Postgraduate Affairs, Faculty of Pharmacy, Girls, Al-Azhar University, and Professor Dr. Samar Ahmed Zaki, Vice Dean for Student Affairs, Faculty of Pharmacy, Girls, Al-Azhar University, And of course, my special thanks to the conference coordinator, to Professor Dr. Iman Hassan. So today, the topic of my talk mainly dealing will be modeling the impact of modeling the impact of meteorological factors on COVID-19 spread and transmission in Egypt. So, in December 2019, a novel coronavirus disease, which known with COVID-19, was reported as an epidemic in Wuhan, China. And also, after the huge number came to the world, on March 2020, the World Health Organization, WHO, has declared the novel coronavirus as a pandemic. Since this time, the spread and transmission of this virus is very high and quick movement across the world. So, as approved by many scientists, that weather conditions play an important role in the spread of different diseases, especially the one related to the viruses. So this research mainly is trying to develop a model to predict the transmission of COVID-19 with different weather conditions across Egypt. So we will to understand the impacts or the relation between the weather conditions and the COVID-19 in Egypt. So, in fact, many statistical calculations and data sets have been used to build and investigate this relation. In this regard, many different meteorological parameters and the coronavirus outbreak over Egypt have been downloaded from the NASA website and from the WHO website. So,
in the past, scientists have found that certain diseases have seasonal cycles. So that begs the question, can the spread of coronavirus be linked to distinct weather and climate? NASA's Earth Observatory says scientists are studying environmental data to hopefully figure out how it will behave. Check out this animation showing NASA models of temperature and humidity, two variables that could be linked to the virus's spread. But they're not stopping there. They're seeing if it could be linked to seasonal shifts like rainfall, winds, soil moisture, and more. To do this, they're using NASA reanalysis data sets to bring together different info into a coherent look at what's going on. NASA says they're creating consistent four-dimensional snapshots of Earth's atmosphere and surfaces. One reanalysis product makes 5 million observations to see the atmosphere at any given time. While they don't have definitive solutions yet, scientists hope to uncover the answers with this detailed data.
Okay, next slide. Of the Egypt map to show you the location of Egypt. Where is Egypt, and what are the main conditions also related to the weather or the climate affecting the area, because this will be very important to understand more about the distribution of this of the climate and weather conditions over Egypt and its relation to the transmission of COVID-19. So this slide shows the annual climate of maximum temperature, mean temperature, relative humidity, minimum temperature, and dew point temperature. And as we'll see, different climatic conditions could affect the Egypt. So this is why it is very important to understand the relation or the impact of this weather conditions on the COVID-19. So this data is mainly from this is observed data from 1983 to 2006. So data and methodology. Here we downloaded different meteorological data, weather data. It started from February 15, 2020 to August 6, 2020 for different meteorological parameters. Mainly downloaded from NASA website, and the website is here. You can see it is free of charge, so anyone can download the data easily for any part of the globe or any region of the globe, or as a point or something like that. So the selected meteorological parameters, mainly it will be shown in table 1 in the next slide. So these parameters are temperature at 2 meters, air skin temperature, dew or frost point at 2 meters, maximum temperature at 2 meters, wind speed at 2 meters, meter per second, the unit, precipitation, millimeter per day, wind speed at 10 meters, meter per second, and the insolation or the solar radiation or sky insolation incident on horizontal surface in megajoule per square meter per day, surface pressure, minimum temperature at 2 meters in degrees Celsius. All temperatures are in degrees Celsius, and relative humidity at 2 meters as a percent. So the COVID-19 also data, in fact, has been downloaded also from WHO website, and this mainly free data. So as we mentioned before, we used many statistical calculations to build this regression analysis. This regression analysis mainly will identify the relationship between the COVID-19 outbreak and different meteorological parameters. So this is our the empirical regression model. We gave this is the formula, and we're and empirical means it is mainly based on the measurements for the whole data, even the meteorological data or the COVID-19 data. So y equals A0 plus A1 x1 plus A2 x2 plus and so on until reach AI xi. So y here represents the outbreak or number of COVID-19 outbreak. A0 is a constant and equals 0 and non-zero in this study. So our calculations mainly based on the 0 for A0 and non-zero for other cases. So we built or we developed two equations so anyone can build easily with the prediction or this empirical multiple regression model. And mainly, so A1 until AI mainly referred to the model parameters, and it will be given after the calculations. Also, X1, X2, and Xi represents the different meteorological parameters like temperature, humidity, and so on, or solar radiation. Also, the confidence interval was set to 95% during this study. So this is table of metrological parameters. So we don't need to repeat it again, and so it is easy to recognize the metrological parameters and the definition and the unit of each parameters. So results and the discussion in this case, because we have a very limited time, 15 minutes. So I'm trying to finish within time. I have to respect time and respect other colleagues to present their work. So as you see here, this graph gives the temporal pattern of COVID-19 daily outbreak in Egypt from February 15, 2020 to August 6, 2020. As you see here, and the horizontal axis represents the date, while the vertical axis represents the outbreak. And as you see here, this is the wave started from this date, and mainly the peak is focused and mainly occurred from the end of May to the first week or second week of July. Then
excuse me, moderators, can you run the presentation till the conclusion, please? I'm sorry, Dr. Iman, the moderator, can you run the presentation for the recommendation, please? Because time is going, we are passing our time.
Temperature.
Yes, dear Professor Gamal, please, our time is very limited. Our time is very limited. So please run to the conclusion, please, Professor Gamal.
Here outbreak total is slightly smaller than the one with constant zero than the constant not zero. So the outbreak in this case with constant not zero is a case of July 2020 value, even positive just like is a case of the relation between the outbreak and the temperatures, or negative like the relative humidity. So the COVID-19 outbreak has been inversely proportional to the relative humidity, while it was directly proportional to different temperature types. The temporal pattern of the COVID-19 outbreak in Egypt was in harmony with different types of temperatures. So the highest positive experiment correlation between the COVID-19 outbreak and the meteorological factors was recorded for the Earth's skin temperature or surface skin temperature. Then it is followed by the mean temperature, maximum temperature, and minimum temperature, respectively. So we will be able to develop an empirical models to predict the transmission of COVID-19 with different weather and condition across Egypt. At the end, I would like to tell everyone that we are very open to collaborate with anyone in Africa, because I am African. So I proud that I am Egyptian and African at the same time, but I'm working in the United States. But in the same time, our university is very open. Our college is very open to collaborate. We are very happy to find any opportunity for collaboration. Okay, so thank you. I would like to thank you all very much for your time and attention. And if any questions could be directed to my email, and I will be happy to answer at the same time. I consider myself as a good communicator, so I will reply maybe within minutes or seconds or something, depends on the time, because the time difference. Thank you very much, and have a nice conference. Be safe, please, and I hope Africa will be great all the time. Thank you.
Thank you very much, Professor Dr. Gamal, for your excellent presentation. and very interesting information. Thank you. And sure, if there is any questions, we will send you, and I'm sure you will be pleased to answer. Now we will move to the end of the current session with the most famous character, and I'm very happy to introduce her, as I lost this opportunity to thank her from the start, and now I got such opportunity to thank you, dear Professor Aman El-Sharif, chairperson, and behind every success. No words enough to thank you. And now it's my pleasure to introduce you Professor Dr. Aman El-Sharif, Dean Faculty of Pharmacy for Girls, Al-Azhar University, Regional Coordinator of Regional North Africa Office of Association of African University. She is Professor of Microbiology and Immunology, member in National Committee of Women in Science, and the former Director of Training Sector in National Authority of Quality Assurance Accreditation of Education. She is also a member of the Higher Committee of Strategies Planning, Al-Azhar of Al-Azhar University. Internationally, El-Sharif is an international expert in the field of quality assurance of higher education. She is member of a high quality assurance project funded by European Union and African Union to establishing a continental African standards guidelines and quality assurance consultant in the American Accreditation Council for Pharmacy Education. She is also a DAD alumnus and founder of different public health awareness initiatives. Professor Amani is a reviewer and editor in different peer-reviewed international scientific journals and chosen as editor of International Journal of Cancer Immunology and Immunotherapy. I'm very happy, Dr. Amani, to introduce your talk, which entitled COVID-19, a portal for back to nature approach. Unfortunately, after what happened.
Thank you, Professor Samah. Thank you. Thank you very much. And I'm so sorry for all attendees for extension of the last session. But as we promised in the beginning of the conference, that the winner of the award for excellent research have got the credit of giving his talk about his presentation or her presentation live. So I dedicate my time of my presentation for Dr. Mona Kamal. And after that, I will come back for you for the recommendation and for the group photo. So I'd like to introduce the winner of Research Excellency, Dr. Mona Kamal, to give us a hint in very short time about her presentation. And for my presentation, it will be uploaded on our website, so you can watch it at any time at your ease after closing the conference. Thank you, Dr. Mona. Welcome, and the platform is yours.
Thank you, Dr. Aman. Can I share a screen? Bismillah ar-Rahman ar-Rahim. Hello, my listeners. It's of my pleasure to participate in this conference.
Dr. Mona, as I usually advise you, raise your voice, please, and share the screen. You are a co-host now. You can share the screen and open your camera if you would like, but
yes,
kindly raise your voice.
Now. I share screen now?
No, not yet. Not yet, no. يا جماعة حد يساعدها، انتوا مش قلتوا جربتوا معاها؟
Okay, doctor, it's clear now.
Dr. Mona, your voice, you should raise your voice, yes. Please.
Okay, thank you.
More. About three times more.
Bismillah ar-Rahman ar-Rahim. Hello, my listeners. It's of my pleasure to participate in this conference by a scientific PowerPoint presentation and present it as a member of the Authors Committee from Al-Azhar University in collaboration with Cairo University. The title is Enhanced Oral Bioavailability of Paclitaxel with B-glycoprotein blockers, Smart Extraction, Anti-Tumor and Side Effect Study in Mice. Introduction. Paclitaxel, which has a trade name Taxol, is a plant alkaloid, mitotic phase-specific anti-cancer drug, which inhibits microtubule depolymerization into tubulin protein, thus inhibiting cell division. Paclitaxel has poor water solubility, so dissolved in Chemo-4L and ethanol for IV dosage form. Paclitaxel has poor oral bioavailability due to the presence of P-glycoprotein efflux pump in the intestine, which efflux in xenobiotics and cancerous agents. Aim of this study, an attempt to enhance the bioavailability of oral paclitaxel administration through the use of B-glycoprotein blockers, either cyclosporin or verapamil, and study their bioavailability, toxicity, and anti-tumor activity. Advantages of paclitaxel oral administration over its parenteral use, including medication no longer require a visit to the outpatient clinic, better maintenance of therapeutic plasma levels, also avoiding skin hypersensitivity reaction caused by extravasation of Taxol Chemo-4L vehicle. Methods and results. Experimental design. Female Swiss albino mice are divided into 4 groups: intraperitoneal paclitaxel only, oral paclitaxel only, oral paclitaxel plus verapamil, oral paclitaxel plus cyclosporin. Parameters investigated in this study: for pharmacokinetics parameter, P-glycoprotein efflux pump activity in the small intestine, toxicity study, total leukocytic count, cardiac toxicity study, by determined biochemical tests, lactate dehydrogenase, creatine kinase MP, and histopathological examination of cardiac tissue. Anti-tumor activity, which determine the inhibition of tumor growth. Animals were subsequently sacrificed at interval times starting from 0.25 minutes till 24 hours after injection of paclitaxel, either oral or parenteral. Paclitaxel extracted, then analyzed by HPLC analysis. The choice of the organic solvent for extraction of paclitaxel from plasma samples has several pilot experiments where initially tried out to find the most suitable organic solvent to extract paclitaxel from plasma samples of mice. The tested solvents were diethyl ether, perchloric acid, acetonitrile, and dichloromethane. The results indicated 100% recovery of dichloromethane compared by other solvents. Pharmacokinetic parameters were calculated by non-compartmental method using software package top fit, using this equation for bioavailability. Pharmacokinetics results indicate that area under the curve of oral paclitaxel with verapamil or cyclosporin compared with intraperitoneal paclitaxel only indicated that the Cmax, Tmax, and area under the curve. Oral bioavailability of paclitaxel resulted in 9.4% of intraperitoneal cyclosporin enhanced the bioavailability to 54.4%. Paclitaxel with verapamil increased the bioavailability to only 25.3%. Assay of B-glycoprotein using rhodamine 123. The method depends on measuring the fluorescence of rhodamine 123 dye, which is transported by the membrane efflux pump B-glycoprotein in the same way as paclitaxel in the intestinal cell membrane. The results indicated that
Dr. Mona, do you have a problem in the voice?
No, I'm with you, doctor.
Dr. Mona, can you raise your voice?
Okay, thank you. Okay.
You have 1 minute.
Sorry, doctor.
No.
You have 1 minute, dear
No.
Dr. Mona. You have 1 minute.
Okay. The result for lactate dehydrogenase and creatine kinase MB indicate significant increase in verapamil than cyclosporin, so confirmed by myocardial histopathological examination. Anti-tumor activity indicate the percentage of inhibition of cyclosporin significantly increased in tumor growth inhibition than verapamil. Conclusively, oral bioavailability of paclitaxel enhanced by 54% with cyclosporin than verapamil, which alone only 25%. Cyclosporin increased P-glycoprotein blocking activity than verapamil. So verapamil also worsens the toxicity of cardiotoxicity parameters. Combination of cyclosporin or verapamil with paclitaxel doesn't affect its anti-tumor activity. Development of smart sample method of paclitaxel extraction from plasma samples, which could eventually be applicable for routine monitoring of the drug in patients being treated with the drug. Thank you.
Thank you, Dr. Mona, and congratulations again. And now let me, in few minutes, share our recommendations set by all the speakers and the moderators during the 3 days of this very rich conference. And first, I'd like also to remind you all to be ready for the group photo of the conference, and for our conference recommendations collected during the 3 days. Here I will announce it on behalf of Professor Mahmoud Siddiq, the Vice President of Al-Azhar University for Research, Postgraduate Studies and Research. This conference was to support healthcare heroes, and despite the current challenges caused by COVID-19, we figured out our responsibility to development as healthcare professionals to gather global efforts and ensure their sharing and availability to support healthcare heroes in the first conference. And our recommendation include within the first track, which under the title of healthcare support, is first recommendation to establish proactive health policy to forecast future pandemics with development of standard protocols for handling future pandemics. Second one, increase the pool of critical and intensive care staff using other healthcare team members and apply national medical registry. For track 2, we have more recommendations. They include building healthcare and the pharmaceutical capacities, establish prediction research center. African universities should invest more in technology, infrastructure, and research, adopt new technologies in education, and also developing mobile apps for diagnosis, tracking of new drugs and delivery system to reduce toxicity and side effect during treatment of COVID-19 or other pandemic. Investment in capacity building in the area of big data and big data analysis, encourage and facilitate multi-institutional based research on infectious diseases through adequate funding and industry academic partnership. And for track 3, establish repurposing of drugs strategy and raise awareness for healthcare waste management, maintain a balanced environment is a human responsibility to prevent zoonotic diseases, formulate regulations for traditional herbal complementary alternative medicine to boost immunity and to protect against infection, and explore what nature has blessed Africa with in terms of phytochemical or phytopharmaceuticals. The big recommendation was to create a global network to gather efforts to face future challenges. So we invite all of you who would like to join our network to just register on our conference, and we will establish a network platform to share all resources, all update works and experiences, not only during these 3 days, but we would like to sustain and maintain our sharing and the network. So if you would like to join us, please fill the registration form. And another recommendation was about holding this conference annually, and hereby we announce the second International Pharmaceutical and Medical Sciences Conference from 1 to 3rd September 2021 under the theme to ensure healthy lives and promote well-being, which is SDG number 3 in United Nations Sustainable Development Goals. And we hope we achieve it on 2030. So our conference will be a step forward to achieve it. And now this is was for the recommendation, and I'd like to ask, before having the photo, to ask my colleagues to demonstrate the list of the organizing committee, because you have just seen what in front of the screen, but behind the screen there is a big team, and I'd like to appreciate and acknowledge and thank them. And please, Anas, start the names of our organization team, and after that, please get ready for the photo. I would like to thank Egyptian Knowledge Bank for their support. I would like to thank Association of African Universities for their support and providing us with this platform for 3 days and hold all their webinars till we finish the conference. I'd like to thank the Multiple Advance Incorporation in Canada for the generous offer of publishing the articles of this conference, full articles for free in SCOBA cited journals. So any researcher would like to have this opportunity and use it, just send us an email on conference email to elaborate clearly your intention to publish your paper in on these journals, and you have the names of the two journals on our platform. And now I'd like to ask the management team to direct us what to do to get the group photo. Dr. Heba, what we should do to have the group photo? Anas? All the team are here on Teams while we are having the webinar on Zoom. So please leave Teams platform and come back to the Zoom and switch on your camera.
And until that, we need our participants to open their cameras.
Yes, please, all participants, if you don't mind, open your cameras to have a group photo for our conference. We have Dr. Raoula Sayed, the provider of the award for young researcher. Thank you, Dr. Raoula, for your generous offer and for being with us. Dr.
Okay, we are taking the photos now.
No, I
Professor,
think
will
we have
wait
to
all
wait for
opening
about 3
panel.
minutes because the number of screens increase by time. So just give them 3 minutes, please. I think we will have 4 photos maybe because I have 4 screen full of participants. Anas, you are the boss in this moment, so tell us what to do.
We will take screenshot for these photos, I think. So we will start from slide 1.
Okay, do what should to be done, and after that, tell us when you finish. Dr. Abir Qasim, can you open your camera? Yes, please. Okay. When you finish, Anas, please just tell us.
Oh, okay, doctor. Okay. Okay, Prof. Amani.
Did you finish?
Yes.
Okay, thank you for all eminent speakers. Thank you for all moderators. Thank you for the organizing committee and for participants and for reviewers, of course, who did a review for a huge number of research in a very short time. And thank you for Al-Azhar University who supports this conference and make it possible. And thank you for my colleagues in the faculty, and thank you all. And we would like and appreciate if you give us some concern and fill the questionnaire, which is will be here in the chat box, and be generous with us to let us know what is our weaknesses and what is our strengths during this conference to get the good lesson for the coming events. And for the people who will start participating in the workshops from tomorrow, we will start workshops at 5 o'clock p.m. Cairo local time. So you are welcome to come, even if you are not registered. It's for free, and it's 15 workshops in different skills. And also you have all the data on our website. Thank you, and have a nice weekend. And it was a pleasure to having you with us during these 3 days. And I hope the recommendation are suitable for your efforts and meet your expectations, and all the conference activities meets your expectations. And goodbye.
Thank you, Dr. Amani. Thank you very much.
Thank you.
When you're
I
doing
think
photograph,
Anas, you can unmute mics so we can hear some comments from our participants and our colleagues and our professors.
when are you doing the photograph, madam?
What?
When are you doing the photograph session?
Yes, this is the photograph session. Professor Bahat was in the photo, Anas?
Yes.
Is that okay? Yes. We will send you the photo, Professor Bahat.
Was I there in the photograph?
Yes, you were there in the photograph, and we will send for you the photo.
It was wonderful being with you. It was a lovely conference, one of the best I've seen. Thank you very much.
Thank you. Thank you.
All your hard work was
you.
showing. Great. Thank you. Thank you. It's lovely to interact
Thank
with
you.
you. Thank you very much.
Thank you all professors. Thank you, Professor Femi. You was having a high degree of commitment. I think Professor Mahmoud Siddiq is us now, and I think he should give a message from Luxor. He is in a medical mission in Luxor. So Professor Mahmoud Siddiq, the Vice President for Postgraduate Studies and Research in Al-Azhar University, and one of the heroes in the fight against COVID-19. Dr. Mahmoud, are you hearing me? You can give us a few words to close by this session. Dr. Mahmoud?
Everyone cheer us in this meeting.
Can you raise your voice, please?
I would like Dr. Amani, you hear me?
Yes, I'm here. Yes, I hear you. Yes, it's okay.
Hello?
Yes, Prof. Your voice is clear now. Go ahead.
I would like to yes, I want starting to practice this recommendation in future. You thank you.
Thank you, Prof. Thank you. And thank you for being with us in spite of all the challenges to have internet connection. Thank you. Okay. Thank you, Professor Hala Al-Adli, for being with us. It was a pleasure to having you.
Thank you, Dr. Amani, for great conference, and thank you for Al-Azhar University. Thank you for your effort.
Thank you all. Okay. See you in our coming event, and thank you. And don't forget to fill the questionnaire, please. And we invite you, if you would like to be in our global network, to register in the registration link in the chat box.
It will be with my honor. Thank you very much.
Okay, bye-bye. Can you raise your... Okay, thank you, team.
Thank you.
I had the passion to study the Arabic language since I was a kid. Reading about early Arab pioneers always made me feel super enthusiastic about this authentic culture. I got fascinated by the Arabic calligraphy. There was something always pushing me to study this language.
أول حاجة بنتعلمها في كلية إعلام إن صورة واحدة ممكن تعبر عن 1000 كلمة. أهم حاجة وإحنا بنختار الصورة لازم نتأكد إنها تستحق فعلا إن كل الناس تشوفها. الكلية بالنسبة لنا مش مجرد مكان، ده عيلة وأصحاب وأحلى ذكريات.
Mon objectif, ce n'est pas d'apprendre une langue étrangère. Ce qui m'intéresse, c'est plutôt la communication avec les autres. Si nous parlons plusieurs langues, nous allons certainement mieux comprendre le monde qui nous entoure. Étudier une langue étrangère ne m'a pas seulement permis de comprendre les autres, mais aussi de me faire des amis et de trouver de nouvelles familles partout dans le monde.
Pengajian perubatan merupakan salah satu impian yang mampu dicapai di sini. Kami di sini bukan sahaja belajar tentang perubatan, namun kami juga belajar bagaimana cara untuk meraih keyakinan pesakit dalam menerima rawatan daripada kami. Harapan kami, semoga kami menjadi duta universiti ini sekembalinya ke Malaysia nanti. Tempat ini bukan hanya sekadar fakulti, tetapi ia merupakan rumah kedua bagi kami.
What does it mean to study here? One of the oldest universities all over the world. With a glorious past and promising present. 79 undergraduate colleges in different fields. Science, pharmacy, astronomy, radiology, agriculture, architecture, engineering, art, sports, language, and Islamic studies. 46 research centers. $40,000 scholarships for foreigners. Graduates from more than 150 countries all over the world. Welcome to Al-Azhar University.
I had the passion to study the Arabic language since I was a kid. Reading about early Arab pioneers always made me feel super enthusiastic about this authentic culture. I got fascinated by the Arabic calligraphy. There was something always pushing me to study this language.
أول حاجة بنتعلمها في كلية إعلام إن صورة واحدة ممكن تعبر عن 1000 كلمة. أهم حاجة وإحنا بنختار الصورة لازم نتأكد إنها تستحق فعلا إن كل الناس تشوفها. الكلية بالنسبة لنا مش مجرد مكان، ده عيلة وأصحاب وأحلى ذكريات.
Mon objectif, ce n'est pas d'apprendre une langue étrangère. Ce qui m'intéresse, c'est plutôt la communication avec les autres. Si nous parlons plusieurs langues, nous allons certainement mieux comprendre le monde qui nous entoure. Étudier une langue étrangère ne m'a pas seulement permis de comprendre les autres, mais aussi de me faire des amis et de trouver de nouvelles familles partout dans le monde.
Pengajian perubatan merupakan salah satu impian yang mampu dicapai di sini. Kami di sini bukan sahaja belajar tentang perubatan, namun kami juga belajar bagaimana cara untuk meraih keyakinan pesakit dalam menerima rawatan daripada kami. Harapan kami, semoga kami menjadi duta universiti ini sekembalinya ke Malaysia nanti. Tempat ini bukan hanya sekadar fakulti, tetapi ia merupakan rumah kedua bagi kami.
What does it mean to study here? One of the oldest universities all over the world. with a glorious past and promising present. 79 undergraduate colleges in different fields: science, pharmacy, astronomy, radiology, agriculture, architecture, engineering, art, sports, language, and Islamic studies. 46 research centers, 40,000 scholarships for foreigners, graduates from more than 150 countries all over the world. Welcome to Al-Azhar University. What is your dream?
I dream of education that unifies nation, creates better communication, strengthen the relationship between me and my friends, gathering rather just disbanding, spreading spirit of love and brotherhood among my kids' generation.
I do have a dream. I am a teacher. I would love having more STEM curriculum focusing on 4 disciplines: science, technology, engineering, and mathematics. I would love to hear that the number of teachers who are qualified and proficient in dealing with these subjects is increasing.
And what about you? What are your expectations for better education?
I expect a better education that can break girls free from customs and traditions. It will help the society better understand girls' roles and their ambitions, while allowing women to occupy leadership positions that give them the chance to make a difference in their communities. It will also allow men to perceive women as partners rather than followers.
And you? What do you dream of?
Well, people of my country have suffered from the armed conflict and other forms of wars. It caused a lot of havoc in the communities, with millions being displaced from their original